Hello, good afternoon. First of all, I would like to thank BGRI for giving me this wonderful opportunity to present my work in such a huge platform. So thank you. Without delay, I would just like to go over my presentation. So as we are all aware that stem rust pathogen primarily infects cereal crops, wheat being one of the most important, and that's why we are here. But the story of the stem rust pathogen do not end in this primary host. The pathogen moves to completely unrelated plant called Barberry to complete its sexual cycle. And Barberry is very important to stem rust. Diane mentioned a little bit in the morning today. The sexual recombination that's happening in Barberry allows pathogen to maintain genetic diversity. And today, in my talk, I'm not going to go into that direction of how Barberry plays an important role in generating new virulent races. Eugene and Anna will be discussing more of that aspects. However, today, I would like to briefly discuss how we have been controlling how we have been dealing with Barberry because of its connection with the stem rust. Because Barberry was known as a culprit for many stem rust epidemics in the history, we actually carried out the eradication program, and the eradication program can be traced back to 1600s in France, and there were many happening in Europe during 1800, but the most notable one was in the US, in which more than 100 million Barberry bushes were destroyed. And again, this morning we heard from Diane that Barberry is coming back, and I believe Eugene and Anna will be discussing more of those. However, today, like Iago said, I am going to give completely new perspective, and by introducing this evolutionary concept that the modern day macrocyclic heterogeneous Puccinia we have today is evolved from the progenitor that existed in the acial form, infecting dicot plants that belong to Barberry even prior to its expansion to the grasses. So the point here is Barberry has been dealing with the stem rust long time ago than wheat has. So we are asking, can we actually learn something new or some new ways to cope up with this pathogen by looking into this evolutionary relationship in between Barberry and the stem rust? And just to tell a little bit more on Barberry, Barberry is actually very highly diverse genera. There are more than 500 species, and this is present in all 6 continents except Australia. And these are the few diversity that you see in Barberry. This is only a few. Again, now coming back to Barberry and its connection to the stem rust, not all Barberry species within that highly diverse genera are susceptible. And in our lab, we are particularly using 2 species of Barberry. The first one is the common European Barberry, which is a typical textbook susceptible Barberry, and this is the one which have been targeted to the eradication program. The second one is the Japanese Barberry, which is a typical textbook non-host, and this Barberry is extensively used in ornamental industry in US and Canada. So in our lab, we are asking a very simple question: what makes this Japanese Barberry resistant, but not the European Barberry? And in our regions, we have this another species, the interspecific hybrid in between this susceptible Barberry and the resistant Barberry, and we are asking these questions: can we use this hybrid Barberry to actually understand the non-host resistance in Japanese Barberry? But there are so many things unknown about this system. This is really cool, but we have no idea if this hybrid Barberry segregates for rust resistance, and that's the first question that we needed to answer. So in order to answer that, we decided to focus on the natural accessions of the Barberry. And in some part of the New England, where I come from, we found a site where we located all 3 taxa in the regions. And we did a field survey first to know how commonly are Barberry distributed into the region, but most importantly, we wanted to collect a population so that we can ask that important questions if hybrids segregate for rust resistance. And the picture here is a map of a site, and these blue pins that you see are the Barberry that we located in the site and we sampled. And within that small site, which was about 6 hectares, we actually located 1,000 Barberry. It was predominated by Japanese Barberry, but there were a significant number of European Barberry and the hybrid Barberry. We then phenotyped, we just collected these plants from our site, we propagated them, and we phenotyped them at the Cereal Disease Laboratory by controlled inoculations, and also in the University of New Hampshire. And one thing I would like to point here is we don't have very standard protocol for the Barberry testing program, and we had to use the naturally infected grass from Elymus repens as our inoculation source. And we were lucky. We actually saw the segregation. And probably you're thinking I'm the craziest one because nobody get excited because of the stem rust happening in Barberry. But this was not the whole story. What we saw was in the hybrids, there are a whole different array of the phenotype. At first, we only thought there's resistant and susceptible plants. But we saw the different phenotype, and those pictures, you can see some plants with a lot of hypersensitive reactions, some plants without any visual symptoms. And to summarize, we tested 105 hybrid plants, and out of those we found 52% of them susceptible, 35% to be resistant, and about 13% to be the intermediate response. And then we also genotyped those plants, and we found that 53 of those hybrids were true F1 hybrids, and we see the similar kind of segregation. And this result was a little surprising to us because it's not very common for F1 to segregate for disease resistance. So the most important question and the logical question for us to ask is, what is the nature and mode of inheritance of the genes governing non-host resistance in Japanese Barberry? We then started asking the questions by asking if the Puccinia gramineous resistance is cytoplasmically inherited. And we had to ask this because we are working with the natural populations. And in natural population, either of those, the resistant parent could be a female or the susceptible parent could be a female. And if, in the case, if the susceptible parent is female, we'd expect all the progeny to be susceptible. And if the resistant parent is female, then we'll expect all the progeny to be resistant. And as we are working in the natural population, the gene flow occurs equally in both directions, and we'd expect 50% of the hybrids to be susceptible and 50% to be resistant. So that's the rationale, and we kind of saw similar segregation pattern in our population. So that's why we decided to test the cytoplasmic inheritance. And to test the hypothesis, we actually created the control cross by making the susceptible parent as our female. And in such case, we would expect all our hybrid progeny to be susceptible because that's the mother plant. But then we saw the similar segregation that we saw in the natural population. The F1 hybrid actually segregates for rust resistance. And from that, we were able to conclude that the Puccinia gramineous resistance is governed by nuclear genes, not cytoplasmic inheritance. And the genes must exist in the heterozygous condition within Thunbergii for us to see the segregation. But this was good news for us. If the inheritance is nuclear, then we should be able to use this mapping population to do the QTL analysis. So we increased the population size, and in order to do the QTL, we created the linkage map for both the parental species. And at this point, we have a very decent looking linkage map for both the species, and we have 14 linkage group corresponding to 14 chromosomes in both the species. And then we did the stem rust phenotyping, and for phenotyping, we developed 4 classes. The typical, the class 1 is the disease reaction that you see in the resistant parent, where you would see no visual symptom at all or very minor flakings. And the class 2 would be the reaction where you would see a lot of hypersensitive reaction but no development of disease. And the class 3 would be the reaction in which you would see a lot of development of disease, but then you would also see a hypersensitive reaction. And class 4 would be the typical disease response to belonging to the susceptible parent. So now that we had both the linkage map and the phenotype, we were able to run the QTL analysis, and we found one QTL in resistant parent with large effects in chromosome 2. So this was great news for us, but as you see in that picture, it was fairly large region, 13 centimorgan. So what do we do with that? The next logical step for us to do was to build a reference genome so that we could fix the genes in that QTL region. So we actually made a genome assembly. To make a genome assembly, we did a pack biosequencing. We assembled the genome by using the Falcon pipeline. And at this point, we have a very good genome assembly with N50 of 87 megabase. And on top of that, we also did a high-C assembly, which basically orients and scaffolds your context into chromosome. So at this point, we have chromosome level scaffolds, which is 14 again, because that's our chromosome number. And at this point, we have 1.3 gigabyte haploid genome for the resistant parent. And also on top of that, we did a transcriptome assembly, and we used RNA-seq data from 7 different tissues and did a Trinity de novo assembly. And at this point, we have a good transcriptome assembly with 89% of the universal single copy orthologous genes, so which is pretty good for a plant. And then we anchored our QTL regions with our physical map. And our 13 centimorgan in our genetic map corresponded to 3.1 megabase region in the reference genome. And we have 10 contexts belonging to that regions. And through our annotation, we find that there are 239 genes. And now to move forward, we are taking 2 approach, differential gene expression analysis by inoculating the plants and sampling at different time interval corresponding to disease development. And the second would be the variant effect predictor, in which we are trying to do the function-related analysis by aligning the sequence from the susceptible parent to the resistant parent to find the genes associated with the stem rust. So with that, I'd like to summarize. At this point, we have developed a very solid reference genome and transcriptome for the resistant parent. And also, we have reported the linkage map for both the parental species for the first time. And with this, we are confident that we should be able to use this hybrid Barberry to dissect the non-host resistant in Barberry, with the hope that this may tell us new insights, this may give us new insights in the way to cope the disease resistant and also to understand non-host resistance. And for the future work, we are trying to validate the QTL region in the independent population, and we are trying to map these cross QTL regions and characterizing the candidate genes. And with this, this is my favorite quote. I'll just read it. Imagine the benefits if the genes for rust immunity in rice could be transferred into wheat, barley, oats, maize, millet, and sorghum. The world could finally be free of the scourge of the rust. So this is my favorite quote from Dr. Borlaug, and that world would be so nice, but then we won't have this BGRI and we'll be sad, but it will still be a nice world without rust. With that, I'd like to acknowledge my advisor, Iago Hill, and our postdoc, Arthur Melo, who does a lot of bioinformatics work, and a special thanks to Eugene, who have been our major collaborator and a big support for this project. Thank you.